I think we'll go ahead and get started. I think the numbers are still kind of climbing slowly, but it's also a little bit past noon. I'm Stephen Beaupart. It really is a pleasure to welcome everyone here to our Innovation Grand Rounds. This is really a platform and an opportunity for our broader community and our college, our Carl Illinois College of Medicine, to highlight innovators in our community, people that can serve as role models to our students, to all of faculty and staff and others that have really thought about the integration of science and technology as it maybe affects us as humans and in medicine as well. It really is a pleasure to have Professor Gene Robinson as our speaker today. He's director of the Carl Lewis Institute of Genomic Biology. He's also holds a Swanlund Chair here at the University of Illinois. In addition, prior to being director of IGB, he is the director and founder of the B research facility here on campus. He also served as the director of the neuroscience program for about a decade. And his research has led to co-authorship of over 275 publications. Also supported by over $50 million of research funds from NIH, NSF, the Department of Agriculture. And in fact, he was instrumental in getting approval from NIH to fund and sequence the honeybee genome, and really pioneered the application of genomics for the study of social behavior. Other honors that Dr. Robinson has is, in addition to University Scholar, he was named Burroughs Wellcome Innovation Award recipient, received a Guggenheim Fellowship, an NIH Pioneer Award. He is a member of the U.S. National Academy of Sciences and the National Academy of Medicine, and then also recipient of the Wolf Prize in Agriculture, one of the top there. So it's really exciting to have Professor Robinson here to speak to us. His research uses genomics and systems biology to study this mechanism and evolution of social life using the honeybee as a model. And as you can see his title here today, I think it's very interesting how this is a model for how in sociogenomics and how this links nature versus nurture. So please join me in welcoming Professor Robinson, and we look forward to your presentation today.
Great. Thank you, Steve. Thanks for the kind introduction. And it's really a pleasure to be here. I want to welcome the students and faculty and staff and any members of the community. I'm very glad to be here with you and engage with you. The connection to this series is, as Steve said, we use genomics, the technologies that fall under the heading of genomics, to address questions related to the evolution and mechanisms of social behavior that are very difficult to address without these new tools. And what I want to do today especially is go over a new understanding that we have achieved as a result of applying genomics to the study of social behavior, specifically a new understanding of nature and nurture, which has developed over the past 2 decades. So I want to give you some historical background related to this, some of the early studies that we've done to support this new framework, and then talk about more contemporary studies about what some of the issues are as we continue to flesh out the relationship between genes and social behavior. Let's see, advancing, there we go. So let me begin by sort of setting the stage and saying that we are continuing to be at a place in society where it's very easy for us to envision the role of genes in explaining certain behavioral traits, such as athletic ability. And on the top left, you see Archie Manning and his 2 somewhat more famous sons as professional football players. And we see that and we kind of nod our heads, and that all kind of makes sense. But for many of us in society, understanding and accepting a role for genes in other kinds of behaviors, personality, learning ability, is much more fraught and is much more difficult for people to understand. And of course, I'm talking about nature and nurture, the idea that we have over decades, in fact, centuries, polarized this debate and think about genes and the environment. And I must say, when I teach genes and behavior, one of the first things I tell the students is that this field has a legacy of shame to live down. Some of the most destructive social movements in our history have been based on extreme applications of either a perspective of it's all nature, meaning it's all genes, or it's all nurture, meaning it's all the environment. And I list for you some of that legacy right there. However, we now have a new framework, and I will describe that framework to you in the next slide, but I want to set it up first with this slide. This slide shows you the levels of organization in the brain, starting from the highest level, production of behavior, all the way down to the finest or the lowest level. And by high and low, I don't mean anything value laden. I just simply mean levels of organization and the scope of those levels of organization. So to the finest level, the lowest level, DNA, and then everything in between, as you can see. We will be focusing today on the two names that you see circled there, RNA and behavior, and talking primarily about the insights that have been gained by studying gene expression and its connection to behavior. And I want to, in doing that, highlight for you really what I call here the power of the ortholog. So if you look at the scale, there are levels of organization in the brain regions, brain circuits, neurons, and so forth, that are obviously more proximal, closer to the production of behavior. However, in trying to use those levels of analysis to understand evolutionary relationships and understand environment and gene relationships, it's been more difficult for a very simple reason. It's very difficult to look at the brain of one animal and understand how its parts relate to the brains of other animals. This is the case when we go from humans to vertebrates. It's doable, but there's still limitations. And then certainly when we go across evolutionary distance, larger evolutionary distances, such as from vertebrates to insects, it's very difficult to homologize, even to analogize brain regions and certainly brain circuits and neurons. For whatever set of reasons, those levels of structural relationships and anatomy are not well conserved across evolution, or we haven't understood yet the logic of that conservation. By contrast, and so then you will see in the neuroanatomy literature arguments. People might think that this brain region is similar to that brain region in a different organism. Someone else may think, no, it's not, and it's better related to another brain region. And it's very difficult to have clear understanding. By contrast, in genetics, molecular biology, when we go down to the level, we have a concept, which is the ortholog concept. You look at two genes from organism A and B, and if the sequence of those two genes is similar enough with all the appropriate statistical insights and applications, is similar enough, regardless of how diverse those two organisms are, you can say, you know, those two genes are cousins to each other. They're orthologs. And you can do that with statistical precision. And so with that, it is possible to then look broadly across organisms to be able to get the insights that have eluded us in more traditional areas of neuroscience. So we have applied that approach, and that has led to this framework that I want to show you here. First of all, this is the old view dominated by a deterministic relationship between genes and behavior, that there are genetic variables, individuals vary in DNA, in their DNA sequence that leads to differences in gene expression, protein expression, brain structure, brain functioning, and changes in behavior. So that's the old view, hereditary influences on the genome, excuse me. Here's the new view, courtesy of genomics, gene expression analyses, and our ability to move across organisms, and that we see as that social information gets under the skin, affects the brain. There are various neural transduction events. They end up changing the genome, that is changing the expression of genes that then alter behavior and the brain. And hopefully you're noticing the bottom of the slide. Both perspectives are right. We're not replacing one with the other. We are adding the second arrow to the equation. And so then what that means is that it's all about the genome, whether you're talking about nature or nurture. Nature is the green arrow on the top. Nurture is the red arrow on the bottom, both intimately involved with the genome, just over different timescales. The changes that we see on the top, that lead to individual variation, occur from generation to generation and are on the timescales of evolution. The timescales over which the environment affects the brain are quicker. They are moment to moment, weeks, days, months, within the lifetime of the organism. So then we have this interplay between genes and behavior. So that's the main message. If you're busy, you need to go. Genomics has been used to solve the nature-nurture problem by bringing in the dynamic nature of the genome. If you want to stick around, I will tell you about some foundational studies that led to this perspective, and then, as I said, some new issues. So here are the roots of sociogenomics, the subfield that I founded to encompass these multiple roles of the genome on social behavior. So there were two foundational pieces. One, E.O. Wilson's publication of the book Sociobiology in 1975, in which he simply asserted that social behavior must be influenced by genetics and must be shaped by evolution as a biological trait. And the book is considered a landmark in bringing together the study of social behavior across different organisms. It's also was considered very controversial at the time for asserting things about genetics and social behavior that science yet was not able to address. And I'll come back to that point a little later. And then secondly, from a sister field, the study of development, animal development in particular, and the subfield of evo-devo, came the insight, beautifully described in Sean Carroll's book, From DNA to Diversity, came the insight of profound conservation of gene function, that a gene that does X in one species may well be involved in a very similar process, even in a very distantly related species looking elsewhere. And so those were very profound and important roots to the study of sociogenomics. Getting a little closer to home, one of our pivotal studies is listed for you on the right, and pivoted off of a study from the laboratory of Marla Sokolowski, the discovery of a gene encoding a cyclic G-dependent protein kinase that gives rise to natural behavioral polymorphism in fruit flies in the field. This was the first natural polymorphism discovered. This was not induced as a mutant, of which there are many. This was a naturally occurring polymorphism. One version of the gene, that is at the DNA level, sequence differences. One version of the gene gives rise to a more active foraging morph, and the other version to a less active. We looked at this, and this was work that was the dissertation of Yehuda Ben Shachar, who's now a professor at Washington University in St. Louis, and asked whether we might be able to transpose from DNA to RNA, because honeybees also have different behavioral states, some more active than others. And we wondered whether we might see differences not in DNA, but because we're talking about different behavioral states that different individuals express in the same colony, and sometimes they move from one behavioral state to the other. So it's a maturational, a developmental change, not a set in stone DNA change. We nevertheless wondered whether that gene that shows DNA polymorphism might also show plasticity in expression. And indeed it did in the predicted direction. This gave us a huge confidence that we were on the right track. And you'll note the year 2002, for those of you that know the field. This was, genomics was just getting started at that time. Illinois was one of the early adopters of genomics, especially from comparative genomics perspectives. And we were able to take advantage of great infrastructure already at the university to be able to move this from one gene and cloning it and studying it one at a time to genomics. But let me first just give you a taste of the honeybee system and how we are using it for these studies. So I've kind of covered some of these points in passing, but I want to make them more explicit. Honeybees are small, they're insects, of course, but their normal behavior can be easily observed and it can be manipulated. There are many reasons for this, but a foundational reason is that people have been interested in honeybees for a long time, for millennia. Honeybees have provided humans with the first sweetener and the first alcoholic beverage. I need not tell you how important those two categories are. As a result, people have been very interested in learning about bees so that they could manipulate them and get them to produce more honey for their needs. So there's a whole wellspring of knowledge about the natural behavior of honeybees. And indeed, I can assert that aside from humans, the species that we know the most about in terms of its natural behavior on the planet is the honeybee. So this gives us a great resource. Secondly, honeybees display extreme behavioral states. They have a division of labor. Some individuals specialize on one behavior, some on another. This is maturational. As I said, there's an age-related division of labor. Adult worker honeybees live about 6 weeks. They spend the first 2.5 to 3 weeks working in the hive, and then they graduate, as it were, and take up activities that occur outside. These different states they concentrate on, they focus on for extended periods of time. Many animals will move through different behavioral states very quickly, making it hard to study the relationship between gene expression and behavior, because as we'll come back to later, changes in gene expression occur much more slowly than changes in neural firing. And so having these extreme behavioral states, these extended behavioral states, gives us great targets. And so there are bees that act as guards that patrol the entrance. There are bees that act as soldiers to attack intruders. There are bees the ones that you see on flowers that are collecting nectar and pollen. There are nurses that take care of their younger sisters, the baby bees, and so forth. So these are all the tasks that the worker bees perform. In addition, in the hive, there's the queen bee who does not run things, does not tell the others what to do. Her job is to lay eggs. And then there are the males that have very limited activity in the colony, and as far as we know, only exist to mate with virgin queens. So everything I'll be talking about today has to do with the workers. The third point is that honeybees are social, highly social. Everything they do is influenced by their social environment. And moreover, because of our knowledge of bees, as I said previously, we know how to manipulate their social environment in precise ways. The image that you see on the right, those tall white stacks, if you've never seen them before, each of those tall white stacks is a beehive, is a family. It's composed of a single queen and up to 40, 50,000 worker bees. And they all work together as a single unit. we can manipulate that environment. We can make big colonies, small colonies, genetically diverse, genetically narrow, manipulate the demographics, and so on and so forth. We can do that, as the image suggests to you, with large, robust sample sizes. We typically work when we're looking at gene expression or regulatory genomics. We typically will sample 10 to 20 different individuals within a colony, and then from across 2 or more colonies. The image on the left is to point out to you that it's possible to study behavior at the individual level. So previously, giving a talk like this, I would have showed you a slide that had colored numbered plastic tags, which were the state of the art up until just a couple of years ago. Former graduate student in the lab, Tim Gernot, who's now a Ph.D., a postdoc in the lab, developed a system of barcoding bees that has really been a breakthrough, allowing us to do automated 24/7 monitoring of all the bees in colonies to get unparalleled behavioral information. And this will feature in one of the stories today. So these are the attributes of the bee system. Okay, I want to cover 3 principles of sociogenomics. I want to illustrate for you the close relationship between gene expression and social behavior, and particularly how it weaves through the nature-nurture thread. I want to show you the conserved relationship from animals to humans, and then finally explore with you some contemporary issues related to understanding the differences and the ways in which neural control of behavior and molecular control of behavior work together in the brain. So what I want to show you here has to do with some data that we obtained on some of our very first microarray studies. That technique, it was the first method that was used to measure lots of genes at the same time, measure the expression of lots of genes at the same time. It has given way to a technique called RNA sequencing, or RNA-seq, which we are using for the newer work that I'm talking about today. But I want to make a couple of points here. The first point is the table on the top. We compared brain gene expression between nurses and foragers. Here's a fanciful image of that, a nurse bee remaining in the hive, a forager at the flower. And we found that 40% of the genes in the brain show different patterns of expression when we compared nurses and foragers. This is a heat map that just gives you a flavor for the striking differences: yellow up, blue down in terms of regulation. But the point of this table right here is to show you that the differences between brain gene expression between these 2 behavioral states were so strong that it was possible to use a computer program to classify individuals as to whether they are a nurse bee or a forager solely on the basis of the gene expression patterns. And this gives you an indication of the number of correct predictions across 60 possible right answers, so to speak, 60 samples, as a function of the number of predictive genes that were used. You can see then very strong predictive relationship. The table over here gets at a different behavioral state, and that is defense. Here's the entrance to a beehive. There will be some bees that will patrol the entrance. They're called guards. When they sense a disturbance, they release a chemical, an alarm pheromone, that alerts other bees to the danger. The first bees that respond are called soldiers. They fly out and look for the disturbance, look for the intruder, and attempt to sting it. So we have guards, soldiers. Foragers are bees that continue to forage even during a disturbance. And then we have, as I mentioned, the social component, the alarm pheromone, the communication signal that links the guards to the soldiers. This experiment was a nature-nurture experiment. We compared gene expression of guards, soldiers, and foragers between 2 strains of bees, abbreviated here AHB and EHB. AHB are more aggressive than EHB, and compared them to each other across these different behavioral states, and then also looking at alarm pheromone. And what you can see is there are many differences in gene expression associated with genotype. I forgot to mention, we also did cross-fostering. We put EHB bees in AHB colonies and vice versa. That's the nurture part of the experiment. So G refers to nature, E refers to nurture, and what you can see is the answer is yes. There are influences of both hereditary differences, strain differences between these subspecies, AHB and EHB, that give rise to differences in gene expression. Looking at guards, looking at soldiers, cross-fostering also has an effect. We were pleased to see fewer genes affected or differentially expressed in the foragers. That makes sense because there are fewer differences between the foragers of these two subspecies, AHB and EHB. Alarm pheromone had a strong effect on brain gene expression. Here's just a straight up nurture effect, the effect of an environmental signal on brain gene expression. So summarizing these early experiments, close predictive relationship, environmental influences, and in fact, bringing them together, something I did not show you, when we look at the aggressive strain brains and compare the genes that differentiate them from the less aggressive bees, those are overlapping with the genes that are activated due to alarm pheromone. So aggressive bees look like their brain is on alarm pheromone, so to speak. And then cross-fostering, 25% of the variance in brain gene expression is due to the social environment. So some early experiments that lay the foundation. One more early experiment that illustrates the close relationship between behavior and gene expression, even to the point of not one of these very long and extreme behavioral states of guard, soldier, forager, and nurse, but a more dynamic behavior, and that's related to the dance language. So the video that you see shows bees engaging in a very interesting and complex symbolic language that was first discovered by Carl von Frisch, for which he won a Nobel Prize in 1973. It is the only known symbolic language outside of our own. With a series of movements and sounds, the bee is able to communicate distance, direction, and the quality or the value of the food resource located at flowers. So this information provides the ability for honeybee colonies working as an integrated unit to track the changing landscape of flowers. Flowers are among the most ephemeral food sources, very distributed patchily in both space and time. And so honeybees have evolved the ability to overcome those limitations by means of a communication system. Distance, direction, and value, as I said. We wanted to ask whether there are changes in gene expression that relate to distance responsiveness. And here's the experimental setup. These are one of our outdoor enclosures that we affectionately call bee domes. We have 2 of them out at the bee lab. And the reason why we honed in on distance is because of a really great discovery made in the laboratory of Manyan Srinivasan, Queensland University, that showed that the way bees measure distance, so of course, if they're communicating distance, how far a food source is from the hive, they obviously have to measure the distance. The way they measure the distance is via optic flow, the amount of information that flows past their eyes as they're flying to and from the food source. And the way the Srinivasan lab showed this was by training bees to fly through a maze, such as what you see right here. Here's the bee, here's the maze. And what they showed is that if you vary the optical content of the walls of the tunnel, you can get bees to dance as if they have perceived a different distance. So if you have a very dense checkerboard pattern like this, even though the physical distance is short, when they get back to the hive, they will tell their nest mates, their sisters, they will say, hey, I found some great food out there, but wow, it's really a long distance away. And they'll give that information. You take the same bee or bees from the same colony, same food source, same everything, except you change the pattern of the walls on the maze to a very sparse visual pattern. The bees come back and they'll say, yeah, there's food out there. And yeah, it's really close, piece of cake, no problem. They show you those differences. So we used that to be able to do the following experiment. We trained bees. Some bees flew a short distance. This is a schematic, but it occurred in the indoor facility that I just showed you. These bees thought they flew a short distance, and in fact, they did fly a short distance. These bees flew a short distance, the exact same distance, but they thought they flew a long distance because of the checkerboard pattern, the changes in the pattern. And we asked whether any differences in gene expression, trained at the same time, bees from the same colony, and so on and so forth. And getting at this question of how close is the relationship between differences in gene expression and behavior, even for a more fleeting behavior such as this. And the answer is there are differences. There were far fewer. For this study, we dropped down from the whole brain level to looking at particular brain regions that we knew are involved in mediating this behavior. The optic lobes, this is a visual task, measuring distance, obviously. So we looked at the optic lobes, and we also looked at a region of the brain involved in multimodal sensory integration. The mushroom bodies, more about those later. And we found that there was a relatively small number of differences in gene expression between those that thought they flew a short distance and those that thought they flew a long distance in both brain regions. What you can see is a principal component analysis that shows you that even regardless of the brain region, short distance flyers separate clearly from long distance flyers, quote, long distance flyers. Here are a couple of genes that are differentially expressed as a function of whether the bees thought they flew short or long. And these are genes that are known to be involved in neural plasticity. The DUNS is a well-known learning mutant involved in cyclic AMP signal transduction, and GABA, of course, a prominent neurotransmitter. So distance responsive genes in the bee brain, interesting in their own right, but for purposes today to illustrate the close relationship. So the relationship then is not only close, but it's conserved. Let's move on to the second part of the talk. And this part of the talk allows me to introduce and give a shout out to my colleagues at the IGB in the Gene Network in Neural and Plasticity and Developmental Plasticity Thematic Research Group, or THEME, headed by Professor Lisa Stubbs. So this is Lisa right here. The group is composed of Allison Bell. Lisa's an expert in mouse neurogenomics, Allison in stickleback fish, behavior, neurogenomics, and ecology. Saurabh Sinha, professor of computer science, very active in the Carle, Illinois College of Medicine, excuse me, and Dave Zhao, member of the Department of Statistics. And together we set out to address the question that you see at the top of the slide: do brain responses to social information involve evolutionary conserved mechanisms? So what we did very simply is we exposed our three favorite model organisms of the group, mouse, stickleback fish, and honeybees, to salient social cues, either threats or opportunities, looked at their behavioral response, and then looked in their brain to see whether there are changes in gene expression. If you're listening to the first part of the talk, you can almost bet that there were differences in gene expression. But this part of the talk, the question is whether those differences, whether those changes were similar across these very distantly related organisms. So we came up with two paradigms: territorial intrusion, which is salient and a part of the ecology of all three species, and then an opportunity related somewhat to reproduction. In the case of stickleback fish, it's parental care. In the case of mice, alloparental care. And in the case of honeybees, also alloparental care. That is not a direct care of offspring in those two, but in the case of the mice, it was an auntie paradigm. And in the case of the honeybees, these are workers rearing their younger sisters when they're larvae to attempt to rear them to become queens. And workers, as I said, do not directly reproduce. So for a worker to attempt to rear a sister as a queen is as good as it gets. So these are the pretty picture versions of these assays, but in fact, we studied them in the laboratory to be able to control the variables more. These are the pictures on the bottom of the honeybee assay, especially the intrusion assay that I show you. So 10 bees in a dish with an intruder placed in it. So the idea, as I said, expose them to the social stimulus, and then look at changes in gene expression after sampling for those individuals that responded very strongly in the predicted direction to the social stimulus, and then obtaining portions of their brain to be able to measure the gene activity. We relied upon well-known understanding of the vertebrate brain, parts of the brain involved in stickleback fish and mice, involved in responsiveness to social stimuli, such as the hypothalamus. And then we also looked at the mushroom bodies in the honeybee. We reasoned that since it's involved in multimodal sensory integration, learning and memory, as I said before, that it would also be involved in social responsiveness. And in fact, it is, and we've now added that to the portfolio of the mushroom bodies. What did we find? Were there genes that overlap? Are there the same genes using very rigorous methods for determining orthology, that is similarity across evolutionary distance of particular genes? And that's illustrated by the panel, top left panel, which I will not be going through. It's just there to remind me to tell you that there was a lot of very careful work by former postdoc Mike Saul, working under the direction of Saurabh Sinha and Dave Zhao, to be able to establish the bioinformatics to rigorously assign ortholog genes across these very distantly related species, to allow us to ask the question in rigorous terms. Were there genes that responded differentially to social challenge? Yes. Are many of them, or are there a statistically significant overlap of genes across the three species? The answer is yes. I'll give you just a few highlights in the other parts of this image. So on the right, top right, we see some gene ontology categories, biological process categories that were statistically enriched in all three species. And then on the bottom, transcription factors going by their mouse names, but that had honeybee and stickleback orthologs that were differentially expressed. And I needn't tell you that if you see common differentially expressed transcription factors, then that hints at that's the tip of the iceberg. They're involved in orchestrating networks of genes. And so we see then some striking commonalities. Many differences also. A bee is not a fish or a mouse, but some strong conserved features, changes in GPCR signaling, changes in metabolism, as well as other themes that are illustrated for you. So that is a very quick message about the level of conservation across these three species. I also want to show you how we've extended this to humans, taking advantage of an observation that was made by former postdoc Hagai Spiegler in the course of carrying out the honeybee studies. Let me set this up for you with a quote from the book that I mentioned at the beginning of the talk, E.O. Wilson's Sociobiology. It is the task of comparative sociobiology to trace these and other human qualities as closely as possible back through time. This is an assertion of the commonalities that I said was beyond the scope of science's ability in 1975 and led to actually a lot of controversy about both scientifically and socially the implications of asserting this. We now have the tools to be able to explore this using comparative genomics and the concept of the ortholog, as I said, but with important caveats, as I will end this part of the section of the talk with. So here was the observation of Hagai. So here we recall we looked at social challenge. We also looked at social opportunity, which, by the way, I did not show you data about in case you're wondering, just for the sake of time. The story is the same, overlap of genes for opportunity. But when Hagai was conducting the assays, he noticed that some bees read the textbook. They responded to the intruder like they were supposed to, like many, many studies before had shown this. But other bees were ignoring the intruder. Anyone who studies behavior, I guess human behavior too, all too sad an observation, knows that not everyone's on the same page. Some are less responsive to certain stimuli. So he noticed that, and that's something anyone that ever studies behavior will just notice, and we didn't then think much of it. A year later, Hagai was doing the social opportunity assays, looking to see which bees are responding to the queen larva, and again, noticed that some bees are unresponsive. This time, we discussed this, and we wondered whether there might be some bees that are socially unresponsive, or at least less responsive to stimuli. We noticed this separated by 1 year. These are different bees. So it just, this observation sparked the question that was then pursued with more rigor by Hagai and also another study. And so what Hagai did was he repeated and redesigned the two assays to be able to test the same individuals for both stimuli on a repeated basis to ask, are there bees that are not responding to these two stimuli? On the left, you see the ethogram that shows a variety of categories. I want to highlight the red, the blue, and the green. The red were those that responded preferentially to the threat and not the opportunity. That makes sense for bees. They're more like guard bees. That's what we're calling them here. There were other bees that responded more to the opportunity and very little to the threat. These would be nurses. We understand that in bee biology. Guards don't nurse much. Nurses do not engage in guarding, so that fits. Just for completion, you can see that purple group at the very end responded to both, and we're like, wow, that's interesting, but we didn't pursue that. But the key group here, of course, is the green group, the group that did not respond to either stimulus. They had several opportunities to respond to each one. They were tested, repeated two times for each behavior opportunity, and they did not respond. So behaviorally, it looks like there's some unresponsive bees. As you can imagine, following this talk, the next thing we did was measure gene expression in the brain. And I'm not showing you the primary data. This is a treatment of the data known as a principal component analysis. And the point is that when you look at it at this high dimensional level, you can see that the groups pretty much sort themselves out. That is, the red, either triangle or circle, representing two different colonies, are clustered together. That is, the guards, the nurses are clustered together, the blue, and most importantly, the green unresponsive bees. So from the point of view of behavior, they look like a thing in this lab assay. From the point of view of brain gene expression, they look like a thing, like a group. But we wanted to push this a little further, and that we can take advantage of the barcoding system that addresses an entirely different behavior than guarding and nursing, and that is an interaction among individuals to share fluids. So this is the so-called trophallaxis social network. Trophallaxis, a technical social insect term for the sharing of a fluid between two individuals. It's nutritional, it has signaling molecules, and so it's a very important component of kind of like the social glue of a bee colony. So here's the setup for the barcode system. You see the two circled in yellow. They are engaging in this behavior called trophallaxis. Camera, infrared lights, bees don't see, infrared, red light, and so infrared can be used to illuminate an environment and then not disrupt the behavior. This is what the observation hive looks like, a small hive where all the bees have barcodes. So every single bee can be monitored 24/7 for the entire duration of the experiment. And when one does that, one can map out a social network. And here's one depiction of a static social network that just looks at the number of interaction partners and the number of interactions that bees have. I could put it more familiar to you to say this is a Facebook for bees, and these are the number of friends and the interactions among friends. And so what you see is in the center, the queen bee has the highest number of interaction partners and the highest number of interactions. That's sort of a ground truthing. We expect that based on the biology of the honeybee colony. So that's as background for you. But look at the difference between the green bee, the party bee, and the yellow bee. In the same colony over the same period of time, you have an individual that has almost as many interactions and partners as the queen, but then another individual really on the periphery of the social network. This is not a physical network. This is a social network. And you can see just very few interactions. So coming at this from a different perspective, we see that there are bees in a colony, not just in the artificial environment of a laboratory assay, but in a colony that also have very few social interactions. So it does look like social unresponsiveness, whether it's complete unresponsiveness or a much lower level of social responsiveness, that really does exist in the honeybee colony. So we then wanted to ask the question, oh, sorry, one more. I forgot one more point, the most recent information about this, and that is to look at different groups of bees engaged in different activities. This is the work of Adam Hamilton, bringing together division of labor studies with social network studies. And you can see that the bees that in the laboratory test out as non-responsive in those assays that I showed you before, indeed are less responsive in the network compared to bees that can be identified as foragers, nurses, and called here aggressive, but also can be called guards, as well as bees that respond to multiple stimuli. So another way of showing that non-responders are showing low social connectivity. This led us to ask the question, which is an evolutionary conservation question, and that is, are the genes that distinguish the socially unresponsive bees, I showed you the principal component analysis, are those genes that distinguish social unresponsiveness, do they have anything in common with genes that are related to autism? And the answer is yes. And there are two lines of evidence shown here in this slide. One relates to DNA, and that's the top part of the graph. And this is information from the Simons database that show that, so these are DNA differences. And the genes that show highest confidence for genetic variation, DNA differences related to autism, showed statistical overlap with our gene expression-based differences. And then the bottom shows a gene expression analysis post-mortem from autistic individuals and controls, also showing statistical significance. We looked at some controls to see if these relationships are specific, and so far they seem to be specific. We compared our B gene set and looked at other gene sets from other mental illnesses, do not see the overlap. Likewise, we took the autism gene sets that I referred you to in the previous slide, looked at other B behavioral gene sets, and did not see the differences there. So as best as we can tell, the connection, the overlap is between the sets that I showed you, the autism gene sets and the honeybee sets. Now, in leaving this part of the talk, let me be very clear about the following point. Bees are not little people and people are not big bees. I do not want to be glib about autism. Autism is a very complex, serious, multidimensional condition that includes as one component social unresponsiveness. We're not saying that bees are autistic. We're asking whether there are foundational building blocks of social responsiveness that we have extended across vertebrate and invertebrate animal species, whether we can extend that also to humans. And the answer is yes. And so I illustrate this point, I make this point with this image in front of you, a phylogeny, a high-level view of the evolutionary relationships between organisms. And you can see that the last common ancestor for the lineages leading to the humans on the right, and the last common and the lineages leading to social insects, including honeybees on the left, lived some 600 million years ago, is thought to represent a marine flatworm with very simple nervous system, not even yet a centralized brain, and as far as we know, no social behavior. So we're looking at a form of molecular conservation that really has to do with building blocks that are then used and reused during separate evolutionary instantiations of a social brain. Okay, the third and final part of the talk, and this will be much shorter as our time is coming to a close, relates to this challenge that we have that really came out of the finding that, wow, there really is a close relationship between gene expression and behavior, yet genes are further away from behavior than neurons. So how do we understand this? And this framework of neurons are for today, genes are for tomorrow, gives us a framework for understanding the causal relationship between changes in gene expression and behavior, and just beginning to get an understanding of how changes in gene expression interact with changes in neural activity. So let me set this up by showing you that when you disturb a honeybee colony repeatedly every 30 minutes, that's the x-axis, you will, for the first 3 exposures, get an increase in responsiveness and then keep that high level for a sustained period of time. What I just said in scientific nerd terms, every beekeeper knows, which is if you've disturbed the honeybee colony, you should stay away from it for a while because it's going to be riled up. Ecology predicts that exposure to threat in the past often will predict exposure to threat in the future. Honeybees respond this way. Many organisms respond this way. Humans respond this way. PTSD is a condition where the threat never goes away. It's there, embedded, biologically embedded in the brain. So this is a very deep principle that scales very broadly. So what we're finding is that this notion of past predicts future, we can understand in molecular terms. The schematic that you see, very small in the middle of the image, highlights some of the biological processes that I showed you before related to the responsiveness to social challenge involving nuclear receptors, heat shock proteins, and especially for purposes right now, a change in metabolism, brain metabolism. Simply speaking, there are changes in brain metabolism that are associated with the response to the threat and with maintaining that response to future threats. The way we showed this is with both fruit flies and honeybees. In honeybees, former postdoc Claire Richoff, now running her own lab at the University of Kentucky, looked at the gene expression data. From that, designed a pharmacological treatment to change brain metabolism in honeybees. And when she did that, she was able to increase the aggression in a dose-responsive way using 2 different metabolic drugs relative to control, which is the dotted line here, for bees that were not exposed to a threat. They showed the levels of aggression associated with prior exposure to threat just by being treated with these drugs. Likewise, Drosophila, using genetic tools, genetically manipulating the expression of a metabolic gene called ND20. When this was done and expressed only in neurons, this construct expressed only in neurons, flies became more aggressive even though they had no prior aggressive interactions, showing that the changes in gene expression that we see as a result of an exposure to a social context in turn predispose the bees and the flies to further aggression. If you're going to have this kind of a system where genes are involved in responding to changes in social stimuli, you have to also then expect that there will be genes responsive to positive and negative valence. And in fact, there are. A quick summary showing that you recall we did experiments, social challenge, social opportunity. There are some genes that respond to both. Those would be genes that are social responsiveness genes. But then there were others that responded only to the challenge, only associated with the aggressive response. And then others that responded only to the nursing opportunity, to the affiliative response, and displayed the affiliative response. I won't belabor which ones they are and so forth, but I want to just make that point. And we see this over time as well, 30 minutes, 60 minutes, 120 minutes, to give you a feeling for the fact that, yes, neurons respond immediately. Genes take that information and integrate that and cause changes in behavioral state that will last for longer periods of time. So we have the beginnings of this understanding, but really we have to integrate this with neurons. We've been running kind of an end round from RNA to behavior. If we really want to understand this at a neural level, we have to look at neurons. And this is now the work of Ian Triniello, a graduate student in the lab who's in the neuroscience graduate program. And what Ian did was build on this work of Valence in terms of gene expression to ask, are there neurons in the parts of the brain that we're studying, the mushroom body, that are differentially responsive to these stimuli, and can we find them by using some of the genes that are differentially expressed in response to challenge or opportunity. So these are some in situ analyses. The focus was on the mushroom body. I won't go through the anatomy of the mushroom body, but the neuropil region is called the calyx here. The calyces are indicated here, and these are the neuropils here. And what you see is we're measuring the changes in gene expression, looking at in situ analysis. And Ian found that indeed there are subsets of genes in the mushroom body that are differentially responsive to, or that can discriminate valence. Using a gene, two genes, I'll show you, both of them differentially responsive in the previous analyses. The Kenyan cells, class 1 cells, respond to the challenge more so than they do for the nursing assay, and vice versa for the class 2 neurons. Likewise, another canonical gene involved in early immediate responses, EGR, also and differentially active in our previous experiments. Class 1, responsive more to the threat, and class 2 neurons responsive more to the opportunity. So we're beginning to get an integration then. There are neurons that respond to valence. This, by the way, was known in many other systems, but we're starting to connect them to the genes that are responsive to valence. There's some new technologies that are coming online, single-cell RNA-seq and spatial transcriptomics. We're working with Hisan Han, who's an expert and one of the leaders in the development of spatial transcriptomic methodology. She's in the Department of Chemistry and a member of our theme at the IGB. I'll show you, though, some data from the more established, the older of these new assays, new techniques, single-cell RNA-seq, some data that Ian has been generating, working very closely with Professor Dave Zhao. And what you see is that you can, at the single cell, you can find clusters of neurons in the brain that respond differentially to social challenge or social opportunity. Now I have KCs in question mark there because these are dissociated cells. For those of you, excuse me, that know how the method works, there will be dissociated cells of the brain, and we see, and they're united, clustered by their gene expression profile. Spatial transcriptomics will allow us to put those two together. Excuse me. So then, finishing up, what we have are the makings of an understanding of the relationship between genes, neurons, and behavior. The old model, when we think of genes and behavior, was kind of a static model. Genes were far away. They were producing changes in RNA that would lead to changes in protein that would be used as kind of the background, the machinery for the neurons. But instead, we seem to have a more dynamic relationship where changes in gene expression are actually very much associated with the changes in behavior, with the differences in behavior. And so we have then a much more dynamic model. This is obviously a very COVID-dated slide. Who remembers when we were in those kind of lines or calling up an Uber? But anyways, hopefully it still gets the point across with respect to the relationship between genes, neurons, and behavior. Okay, so to finish up, I want to look forward. So first of all, I just want to say that the goal was to talk about some science that relates to understanding the relationship between genes and social behavior. I showed you data to support the idea that there's a close relationship, a conserved relationship, and a causal relationship between changes in gene expression between genes and social behavior. Going forward, I think it's necessary to further extend the sociogenomics paradigm to human. We have to consider policy implications of this dynamic genome model, where it's not all about DNA sequence, but it's about gene activity as well. In order to do that, we really do need new methods to measure human brain gene expression in a less invasive way, whether it's brain organoids, new imaging methods, and we actually are working on new imaging methods. We have a lovely collaboration here at Illinois with the Dean of the Carle Illinois College of Medicine, Dean Lee, Professor Fan Lam, and several other colleagues across campus that are involved in this collaboration to better understand human brain gene expression. And then finally, at a fundamental level, we need to use this information, which mostly was at the level of groups today, and extend it to the level of individuals. We're badly in need of a better understanding of how variation in genotype maps on to variation in phenotype. And the results that I presented today say that a key element of that will be to understand how this travels through the dynamic genome, gene regulatory networks, to be able to put this together. So I end by acknowledging the folks whose work I spoke about. I tried to mention everyone who was particularly important today, in addition to our lab manager, Amy Cash Ahmed, who's involved in all of our work, and former B-Lab manager, Ali Sankey, collaborators, funders, and be happy to take your questions. Thank you very much.
All right, thank you very much, Professor Robinson. Really appreciate that, and really fascinating in terms of just walking us through the sociogenomics, where we're at, and the implications. I mean, there's many implications that this may have in better understanding human behavior and sociology as well. Well, I think we've got quite a few questions lined up in the chat. Maybe I can start with a couple of those, and then please, for the audience, please feel free to unmute yourself and ask a question as well. Let's see. So there was one question that came through. What is known about difference in mechanisms of gene regulation epigenetics between long-term shifts versus short-term shifts? So I believe phase of life versus longer short-term travel. I think this relates to when they were flying through that tube that you described.
Mm-hmm. So yeah, I did not talk today about the ways in which changes in gene expression may be stabilized for even longer periods of time. That usually comes under the rubric of epigenetics. And there are a variety of mechanisms, changes in DNA methylation. Other labs have shown this to be involved. We've done a little bit of work in that area. We have a new project that's looking at the role of microRNAs, another way of stabilizing gene expression, the work of graduate student Sarai Stewart in the program of Ecology, Evolution, Conservation Biology. So those are undoubtedly involved here. They interact then with gene regulatory networks. And we have a multitude of factors that integrate and regulate gene expression.
Mm-hmm. Very good. There's another question about what about energy expenditure differences? Now, I think that was asked before you started talking about metabolism and links to maybe aggressiveness, but I believe that was referring, to some of the earlier comments you were making, like what role does energy expenditure have in this gene expression between, say, the different subgroups of bees?
Yeah, so we're very interested in metabolism, and I have to share a little story there. So when we were looking at differences between related to aggression, we in our earliest studies, we saw changes in gene expression associated with metabolic genes at the top of the list, but we skipped them because we wanted to apply a neuroscience-based perspective. They're not sexy. We wanted to look for changes in neurotransmitters, in receptors, and all the classic neural machinery. And we found those as well, but we returned to those because they were at the top of the list in experiment after experiment. And so changes in brain metabolism are just a very strong and an underappreciated feature underlying neuroplasticity. And so now that we've focused in on them, we're learning more and more about that. And I think that it's just a great example of how genomics allows for an unbiased, discovery-based approach. And then it's up to the investigator to remain unbiased and open-minded and take the data and move with them.
Mm-hmm. Very good. Well, maybe I'd like to open this up for any comments or reflections from some of the clinical partners that we've got on the call today. I see Ken Aronson's name here, but any comments or reflections from the clinical perspective of what we've heard about nature versus nurture?
Hi, Ken Aronson. So terrific talk, Professor Robinson. And as a neurologist and a beekeeper, it hits home in lots of dimensions here. Currently, one of the hives is without a queen. So the pheromone effect and the changes in the orphaned workers is interesting. But I was on a clinical front. You know, we see the evolution in patients from episodic expressions of their biology and their symptom profile and their patterns of disease to chronicity. Things like epileptogenesis, going into a chronic migraine pattern, going into chronic pain with central mechanisms and neuropathic pain. And undoubtedly, these are linked to the types of things that you're describing biologically. And it's very frustrating for the patients, of course, to know how to act, and for us to how to get at the front end of the disease timeline. So these insights are fascinating about what the future might hold for that.
Mm-hmm. Yeah, I think the insight of conservation gives the ability then to ask how do these genes working in networks, what kind of modules or foundational building blocks might there be to be able to understand the neurological conditions. You mentioned chronicity. So there's very well developed biology of timekeeping in the brain. We have one of the world's leaders in this on our campus, Martha Gillette, who has pioneered a lot of work in this area. And as you know better than I do, there's often sleep disorders associated with psychiatric conditions. And so how does this module of chronicity and circadian rhythmicity relate to some of these other behavioral modules? Genes give us tools to be able to explore this in ways that we hadn't had prior to this. And I'm sorry to hear about the queen loss, but I'll give you some advice, but I don't make house calls.
I'll take a queen cell if you have one.
I see we got a call.
Hi, Gene.
So...
This is Mike Hill.
Yeah, go ahead, Mike.
Hey, Mike.
Just a moment.
Hey, hi. Sorry. Just a quick question from a pediatrician. How many people around? So just a quick question from a pediatrician. You're studying an adult, if you will, mice and bees. Is there any difference in maybe some of the results you're going to see with immature species?
Yeah, that's a great question. First, I want to give out a shout out to Mike. He took care of our 3 kids and helped them grow up healthy. And so great to see you. And the answer to your question is that we are seeing more and more evidence of roles of environmental influence early in life. We, of course, have much knowledge of this from humans, but animals, where it's possible to study this, we are seeing very strong influence. And some very interesting work has emerged from honeybees. We did some of it, and a few other labs are involved, that show that even a larva, which doesn't look at all like an adult and has to undergo metamorphosis, complete metamorphosis. The brain is reorganized, the body is reorganized. If you expose larvae to certain environmental stimuli, that will lead, as they grow up as adults, to behavioral biases. So Claire Richoff, someone I mentioned before, did another one of those cross-fostering experiments, but instead of cross-fostering adults, she cross-fostered larvae. She put larvae from an aggressive colony into a less aggressive colony and vice versa, let them grow up as adults, did not let them experience their adult colony environment at all, but removed them to do laboratory assays just on the day they were, quote, born, their first day of adulthood, and there was a behavioral bias there, even though brains have been dramatically reorganized. So we would love to understand that because, as you're suggesting, there are very potent forces that happen before adulthood.
Interesting. Another question from the chat I see that's, this is from Mark Huber. Is there any connection between brain and peripheral gene expression, meaning muscle or circulatory system profiles in these honeybees?
Yeah, that's another great question. There undoubtedly would be, but we've not looked at that. One can imagine that, for example, the maturation of flight ability. Bees, they emerge as adults. I just mentioned their first day of adult life. They can't fly on that first day. They have some structural limitations. They have to mature a little bit more before they're able to fly. One can imagine those maturational changes having to do with the hardening of their skin. Insects wear their skin on the outside, their cuticle, and muscles supporting wing activity would be coordinated with brain activity, gene activity, but we've not looked at that yet.
Thank you.
I maybe have a question too about the subpopulation of bees that were labeling as non-responsive to stimuli, and I think beginning to possibly make a link with autism too. But is that non-responsiveness, is that viewed as being just inherent or instinctual to the bee, or is it a choice, right, that the bee might make?
Mm-hmm. Yeah, that's a great question, especially with the theme of the talk in general. We don't know the answer to that. We don't know whether they are, quote, born that way, or whether they've had experiences that may bias them, or in the spirit of the talk, how both of those factors interact at the molecular level. One plan we have for the barcoding system is to be able to scale this up and run many more colonies at the same time and develop a kind of a predictive analytics using known genome, bees with known genome sequence or sequencing their genomes afterwards, and having complete records of their behavior to be able to build a behavioral analytics platform that could allow us to address questions like that.
Mm-hmm, mm-hmm. Very good. And maybe along the lines of that too. So given that we have a group of students and others on this at this grand rounds thinking about innovation and technology, and so what's needed, right, to next for your studies and to answer some of these questions that you're posing? Is there technologies, is there new science that we need to think about?
Well, I think one area, especially with respect to extending this to humans, is brain organoids, brain imaging, methods that do molecular brain imaging. You're a world leader in brain imaging, so you know some of the limitations that we have in that space, and so we're excited about new possibilities that allow us to not only look at neural activity, but some aspects of gene activity. So that would really be game changing. The brain organoids, both for humans and even for other organisms, also will be game changing. It's very early days for that. My colleagues who are expert in that say that, yeah, it's a brain, but it's a pretty limited brain at this point. And so for the kinds of complex social behavior that we're interested in, it's not ready yet for prime time. But that kind of methodology that would allow us to standardize certain experimental paradigms and compare human and bee and stickleback fish and mouse and other species under the different methods, different conditions would be, I think, really game changing.
Very good. So a question that came in from Professor Mendenhall saying, can you talk about your sociogenomics for your courses and how they can be done on a large scale?
Sure. Hi, Ruby. I'm glad to get your question. So I think that it's possible to take these kinds of insights, for example, and then focus them on, for example, biological embedding, the biology of stress, how does stress get under the system. You and I have talked about a course that we might develop along those lines that takes these insights and looks at that. And so it can trace the action of genes, first of all, the environmental stimuli, how those environmental stimuli are transduced by the nervous system, how do they get embedded, and then cause long-term change in brain behavior, health, and well-being. So I think we have the conceptual framework, and some of the work that I spoke about today hopefully allows one to use insights from animals and then apply them in a suggestive way in a tentative way, in a to be determined type way to humans, but it gives us a toehold into areas that we previously weren't able to go.
Okay. Any other questions from the audience? Let's see, another one. So a comment in the chat, I worked in the B lab in my undergraduate studies from 16 to 18, and now working in the clinical research at Carl. Great to hear an update about your findings over time. In your studies on social stimuli, do you notice any avoidance or aggression from those other bees who are responding to social stimuli toward the unresponsive bees?
Yeah. Well, first of all, hello, and I'm glad you had a good, pardon me?
Jeanette Cullum.
Okay, Jeanette. Well, yeah, great to hear from you. Glad you had a good experience and that you have moved along very nicely. We have not noticed that, and so you're hinting at a very interesting aspect of the work, and that is there any particular role for unresponsive or less responsive bees in the colony? Like, why are they there? What do they do? Why do they exist? There's two possibilities to that. One is that that they don't have a particular role. They exist, that there's some variation in all species in terms of brain and behavior, and they exist sort of on the tails of a distribution of unresponsiveness, but that they're not causing any problems, and so there's no need to get rid of them either in real time or in evolutionary time. The other possibility is very intriguing, and that is that they are less responsive and that they form a reserve force in the colony. And in fact, less responsive individuals have been found in other insect societies and have been called a reserve force. And we have a very nice example of that coming from some of our work. We had a citizen scientist in the lab a number of years ago, Paul Tenzar, who designed a not barcode, but an RFID system to track individual bees in terms of their flight activity. So for those of you from Illinois, and you remember traveling on the highway once upon a time, the iPass system. So, you know, you go under something and you track it. So we had an iPass system for bees. We could track their flight activity. And Paul worked with former Ph.D. student Claudia Lutz and found that there were elite forager bees. 15% of the foraging activity, just based on the number of trips going in and out of the colony, was performed by, over 50% of the foraging activity was performed by just 15% of the bees. So it sounds like a real elitist story, doesn't it? But there's a populist twist. When you remove those 15% who were doing the vast majority of the foraging, other bees who were less active upped their game. They could do it, but they weren't doing it. And so it suggests that there are layers of regulation in a colony because things happen to colonies. Bees get eaten by wasps, by ants, by birds. They get caught in a thunderstorm. Other things happen. A colony can lose its foragers. So here's a group of bees that are kind of hanging back and ready to take over. And that could be the tip of the iceberg. There could be other such reserve functions for bees that appear to be less responsive. So we don't know which of those two scenarios is more accurate, but the second one has some plausibility.
Fascinating. I was curious, you had mentioned, so you talked about these subpopulations of worker bees, right? The forager, the guard, the soldier, the nurse. But how do the gene expression patterns in those bees compare to that of the queen bee?
Yeah, so the queen bee has very distinct pattern of gene expression relative to the workers. There's some, some common things that are involved in basic housekeeping, but there are differences in hundreds of genes between the queen bee and the worker, and same for the drones as well.
Does she have essentially all those traits, those genes, or is it a special population, maybe say a leadership gene, for instance?
Yeah, that's a really fascinating question. The behavioral repertoire of a queen is entirely different than the repertoire of a worker. Basically, queens were named at a time when humans were fascinated by monarchy. If she had been named, if queen bees were named now, and certainly if they had been named by our youngest son, who Mike Hill knows very well, quite irreverent when he was a teenager, queens would be called, hey, yo, egg-laying mama, nothing better than that. They don't deserve any name better than that. They don't tell the workers what to do. They don't have leadership per se. They are egg-laying machines. They produce pheromones that stabilize the behavior of the colonies, but they don't tell the workers what to do at all. So they have a very distinct and very narrow repertoire of behavior. And I'll add that the worker and the queen are derived from the same genome. Workers are females, queens are females. What sets them on different paths in life, it has to do with how much royal jelly they're fed during the first couple of days of their larval life. And if they get a lot, they go down the queen path. If they don't get a lot, they go down the worker path. But the regulatory architecture, the machinery, molecular machinery for these two different lifestyles is there in the genome. And you know, it'd be fascinating to be able to unpack that and understand that, to be able to hone in on particular genes associated with particular traits, like your question suggested.
Fascinating. Any other questions from the group? I think I captured everything in the chat. Okay. Well, Gene, thank you very much again. This is really fascinating. I think really interesting, appealing to a wide audience we had on the grand rounds today. For those attending, you can see in the chat, Jessica Brookabile put information about CME credit, if you'd like to claim that for our clinical colleagues here. But again, thank you. You've now stimulated us to think about bees in a very different light and kind of revisit this nature versus nurture concept. So thank you.
Well, yeah,
thank you all to those attending today.
Yeah, thanks for the invitation. Great to be here. All the best.
Wonderful.
Stay healthy.
Thank you.
Thank you. Have a good weekend, everyone.
Bye-bye.
Bye-bye.